Okay, so we'll add in some anatomy next time. But really want to get to genetics here as quickly as we can to make sure that we get that out of the way. So you are familiar with mitosis, and you're probably going to think that most of this is review, but there's going to be some stuff in here that you haven't heard before. So we used a diagram, or we just wrote a diagram that had the G1 phase, the S phase, the G2 phase, and then we just kind of left mitosis blank. But you're aware that there's a lot that goes on in mitosis that can be put on a diagram like that. And to start with, although we're really told that it's all part of the same process, if we really want to be authentic and faithful to the idea of mitosis, the M phase has two components. It's got the division of the nuclear material, right? We've got the division of the nuclear material, and you know that we call that mitosis. And it's got the division of the cytoplasmic material. What do we call the division of the cytoplasmic material?
Cytokinesis.
Yeah, it's cytokinesis, isn't it? Okay. The word cyto means cell, and kinesis simply means movement. So literally that term means cell movement, which isn't quite what we really mean, right? We mean division of the cytoplasm, but cytokinesis is not literally part of mitosis. It's the division of the cytoplasm. So we can say that these two things are part of the M phase, but cytokinesis is not really part of mitosis. Mitosis refers to the separation of the DNA material. Okay, now there's a lot of information packed in here, and the way that I want you to understand this is going to be fairly thorough. I want you to be able to tell me what's going on in each of these different phases of mitosis, not just in general terms, but also what's going on in the cytoplasm, what's going on in the nucleus, okay? But we can remember all of this stuff in a way that makes sense and that won't involve a lot of memorization, okay? So our starting point is interphase, right? Because DNA is dispersed in interphase, we can't see individual chromosomes. And I think you'll agree, if I look at the actual cell here, I can't see the individual chromosomes, right? They're all spread out. And that's the term that we use. We use this term dispersed to describe what the DNA looks like in interphase, okay? And that's why the early anatomists didn't think anything was going on. They didn't see any action inside the nucleus. It just looked like it was sitting there, and all the chromatin is dispersed, okay? And that means it's spread out, and we need it spread out so that we can actually read the code and make the proteins, okay? Because the DNA needs to be packed very tightly, though, in order to divide and be duplicated, the DNA has to be chunked into individual chromosomes, and these have to become highly condensed so we can actually see them during mitosis. We see them as individual things. In interphase, we see them all spread out, but in mitosis, we see them as these individual things. And you're aware that the beginning of this process is called prophase. What does the term pro imply? So it implies this is what happens before everything else happens, right? Precedes or proceeds. It happens before, yeah. So pro refers to before, okay? Prokaryotes, before the nucleus, right? Eukaryotes, true nucleus. Prokaryotes, before the nucleus. Prophase, before the action actually happens. Okay? What do I have to do to get ready for the action to occur? And I've given you a list of goals and nuclear features and cytoplasmic features along with the diagrams here. But what I said here in this, oh, I didn't want that to happen. So I always manage to move my pictures around when I'm scrolling. Okay, so I said on the diagram of mitosis, identify the set of pictures labeled prophase, and notice the following features. Okay, so here's my prophase. Okay, these are my diagrams referring to prophase. And I basically am creating the situation. I'm setting it up so the action can happen. So in the actual microscopic picture, there is no more nuclear envelope. Let's look at that actual microscopic picture. It's gone, right? I can see a nice boundary around the nucleus beforehand, but now I don't see that. Okay? My nuclear membrane is gone. Okay? This is represented on the diagram by a dashed line. What's the functional significance of the loss of the nuclear membrane? Why do I have to get rid of the nuclear membrane in order for me to be able to separate my duplicated chromosomes? Why do I have to get rid of the nuclear membrane? Yeah.
Then the DNA won't be able to separate into two daughter cells.
But why?
Because the nuclear membrane is too strong for that.
Okay. But what's the relationship between the nuclear membrane or the nuclear envelope and the chromosomes themselves during interphase? What's the relationship between the chromosomes and the nuclear membrane during interphase?
The chromosomes are inside.
Not just inside, they're
Sealed?
They're attached to it, right? The chromosomes are attached to it. If the chromosomes are still attached to the nuclear membrane, they're not going to be able to divide, right? So the functional significance of the nuclear envelope breaking down is the chromosomes need not to be attached to the envelope. I can't have the chromosomes attached to the envelope in order to be able to divide. So I've got to get rid of it. So you don't need to memorize that the nuclear membrane disappears. It does, and you're asked to state that feature, but why does it disappear? I can't have the chromosomes attached to it in order for them to be able to separate, right? Okay, I've already duplicated my chromosomes. I've got them there, but if I don't lose that nuclear membrane, they're going to stay attached to it, and it's not going to work. Okay, so that's one feature of the nucleus during prophase. I've got to get rid of this. I'm setting it up so that I can divide. Second feature, the chromatin is no longer dispersed. It has become highly condensed, and we can begin to see the individual chromosomes mixed together randomly in the center of the cell as large, dark, purple linear structures. Sorry about that. Right? They're randomly dispersed. That's how I know things aren't quite ready yet. This is the beginning phase. If they were lined up or they were being pulled apart, they wouldn't be randomly dispersed, would they? They're randomly dispersed, so it's not quite ready yet. This is the beginning phase. I'm in the prophase. And that's how I tell this is prophase, is that my chromosomes are condensed and they are randomly dispersed. They're randomly set inside this nuclear space here. Okay? And I need to have them condensed in order for me to be able to separate them, right? That's how we go from this ball of fishing wire that would seem impossible to pull apart, right? This ball of fishing line that I can't possibly hope to separate to structures that I can separate. Now I'm condensing them so that I can attach my microtubules to them and pull them to one side or the other. Okay, that requires me to have condensed chromosomes. So I broke down my nucleus. I'm condensing my chromosomes. Okay, so I've asked the same question here. So first of all, the diagram shows the structure of each chromosome. Each structure is actually a pair of duplicated chromosomes, right? We've talked about this before. This is a chromosome, and this is a chromosome. Each one of these arms here is a chromosome. It's not separated like this, and this is a chromosome, and this is a chromosome. That's not the case. This is a chromosome, and this is a chromosome. Okay? So the diagram shows the structure of each. Each structure is actually a pair of duplicated chromosomes that look like two sets of wings attached together at a central point. And what do we call the central point again?
Chromatid.
The central point is the centromere, right? The centromere. Each set of wings is also called a chromatid, although it is actually still a chromosome too. One chromatid will go one way, right? I'll have one go this way, and I'll have one go this way. Okay? And what is the functional significance of the fact that this occurs? The chromosomes condense and become randomly duplicated. I have to, this allows them to be able to be divided, allows, oh dang it, allows chromosomes to be able to be divided. Okay? I have to have them in this condensed state or I can't divide them, right? Okay? But I've said that there's 2 points to this, 2 points of significance. What is the functional significance of this that I've, okay? My brain is sort of freaking out on me right now, and I can't remember exactly what I wanted there as a second point, but I'm sure as I go along, I'll remember what I wanted as that second point, and I'll impart that to you once I remember it. Okay? I'm old. What is the functional significance of this? What is the functional significance of... Okay, I can't remember what I wanted as that second point. Okay? In any case, I can remember that I've got my chromosomes, and all I have to do is just look at the other thing to say, this is not like this at all, right? It's completely dispersed here. They're completely condensed here, but they're still in a random state. They're not quite ready to be divided yet. But I have to do this in order to be able to separate them. I can't keep them in that same ball of fishing wire kind of state. Okay, the duplicated centrioles begin to separate and travel towards the poles of the nucleus, stretching out. Oh, I remember what I wanted. Okay, I told you I would remember what I wanted here. The other functional significance of this, if I've got my chromosomes in this condensed state, can I make proteins?
No.
I can no longer make proteins. My cell is actually at risk at this point because I cannot make any proteins at this point because I can't read my DNA, right? I can't read the code. I can't get a molecule to come in here and say, okay, I need to create this particular protein. Okay, so the other functional significance of this is I can no longer can no longer make proteins. Okay? Once I become condensed, I can no longer make proteins. Okay? So, the duplicated centrioles begin to separate and travel towards the poles of the nucleus, stretching out strands of microtubules between them. This developing structure of the duplicated centrioles and stretchy microtubules is called what? What do we call that apparatus that this creates?
Spindle fiber?
It's the spindle apparatus, right? It's the spindle. Does anybody know what a spindle is? Or what it refers to normally? Yeah, it's like on a weaving machine or a sewing machine. I have a spindle that holds the yarn, right? It holds the yarn, and it has kind of a shape like this. And so the early anatomists thought that this whole structure kind of looked like a spindle, where I have this at one end and this at the other end, the centrioles at both ends, and that this structure looked like a spindle. It's not really, because the yarn is stretching out in the wrong direction here, right? Because on a sewing spindle, the yarn is around it, but in the spindle of the mitotic figure, the microtubules are stretched out longwise, aren't they? Okay? What's the functional significance of this? The machine that separates the chromosomes is being created. Okay, that's the functional significance of this. I'm creating that machine. The machine that separates the chromosomes is being created. I'm creating this machine that's going to pull these chromosomes apart. That's why this is prophase. It's not quite ready yet. It's the beginning phase. It's the before phase. Okay? So that's what we need to say about those things. And it should make sense. I don't have to memorize these features, because it just makes sense that when I'm beginning to get ready to separate my chromosomes, I have to release my chromosomes from the nuclear membrane. Therefore, the nuclear membrane breaks down. I also didn't say this, but if I can't make any more proteins, I don't need a nucleolus. Why don't I need a nucleolus if I can't make any more proteins right now? What's the nucleolus do? What did we say? It makes the ribosomes, right? The nucleolus makes the ribosomes. The ribosomes are the protein factories. Do I need to make protein factories if I can't make proteins? No. So I shut it down. Okay? I need to have my chromosomes condensed so that I can separate them. And then I need to create the machine that's going to separate them. So just by knowing what's happening, I don't need to memorize what's going on in the nucleus. I just know what's going on in the nucleus, because that's what has to happen in order for me to get ready. So that's the beginning phase, or the before phase. During the next part of mitosis, which is metaphase, obviously. And meta means middle. I don't need to memorize anything to tell me what's going on here. If I hear the term middle referring to mitosis, I know that means that all of my chromosomes are lined up in the middle. The arrangement of the chromosomes is no longer random. Each chromosome with its paired chromatids moves to the center of the cell and becomes attached to a spindle fiber at the centromere. Remember that the spindle fibers are microtubules extending from the centrioles, and each side of the centromere becomes attached. So I have a structure that's like this. Here's my centromere, and the centromere is made up of special proteins that are made to lock on to my microtubules. Okay? They lock on to my microtubules, and my microtubules can then pull those chromosomes apart. Okay? Each side of the centromere becomes attached. As the chromosome becomes lined up in the center, they form the appearance of a line of chromosomes. What do we call this line of chromosomes? What do we call this line of chromosomes right here? Anybody remember a term for that? It's called the Metaphase Plate. It's called the Metaphase Plate. When we look at it like this, it just looks like a line. But remember, this is a three-dimensional structure. Okay? So if I cut down the middle here of the cell, and I look at the cell towards the end of the cell, I'm going to have chromosomes all over the place here in a third dimension, right? If I look down on these, I don't just have chromosomes in a line here. I have them in 3 dimensions all throughout that center there. It's like I took a thing of cookie dough, right? If I took a section of cookie dough, okay, and I cut down the middle. If this was the middle of the cell here, I'd have chromosomes stuck in the cookie dough like this. But if I look down on top, if I look down from it on the top, it would look like they're just all lined up in a line there, right? So that's why we call this the Metaphase Plate. This is actually like a plate of chromosomes. So this is 3-dimensional. So Metaphase Plate. And this is so important. This is so important. What does lining the chromosomes up in the middle of the cell like this assure that's going to happen? What does it assure that is going to happen? Okay, a little more specific than that. Yeah, I only have 1 chromosome of each pair going into one cell, right? I have all of the chromosomes going to the right place. Each daughter cell, if I do this, each daughter cell must get 1 of each chromosome. Lining up like this assures this will happen. Okay? Lining up like this makes it so that I can only put 1 chromosome of each pair into each daughter cell. I can't screw this up, right? It won't let me screw this up. The significance of this is I will always get 1 chromosome of each pair into 1 daughter cell. Okay? That's what lining up at the Metaphase plate assures is going to happen. Oh, I probably didn't leave that there long enough for you to write that down, did I? Each daughter cell must get 1 of each chromosome. Lining up like this assures this will happen. Okay, and the process of this, as we'll talk about a little bit later, the process of this is one of the most complicated things in all of biology. The process of how this happens is one of the most complicated things in all of biology, and it's one of the coolest things. So, the only feature I have as far as nuclear features goes is that the chromosomes are lined up at the metaphase plate. I don't really need to memorize that. I can see it. That's the nuclear feature that I'm worried about. And the spindle apparatus is completed. You can almost kind of see it here. You can see little lines. You're not really supposed to be able to see microtubules. And so it's really more of just kind of a haziness there, a suggestion that there's a spindle apparatus. But the spindle apparatus is complete at this point. Okay? So that would be my cytoplasmic feature that I would be thinking of here. Okay? And again, I don't have to memorize things here because the word itself tells me what things mean. Ana means to separate or be apart from. Okay? So if I want to characterize the phase in which the chromosomes move apart from each other, anaphase is a perfect word to use there. It tells me that things are moving apart or separating. And what's going to happen here is that as I have microtubules attached to each of these, the microtubules are going to start to shorten. Okay? They're going to start to shorten from the far end, and as they do that, that's going to exert a force on each of these chromosomes, and it's going to pull the chromosomes apart. Okay? The chromosomes are moving. The fact that they are moving, we can see because it looks like they're caught in a current or a flow of fluid here, right? As I'm pulling the part that was at the centrosome, the tails trail out behind it. So I can see that those chromosomes are moving in opposite directions because of that. Okay? At the same time, we also notice that the cell starts to elongate. Okay? The cell starts to elongate. That's another effect of these microtubules. As these microtubules that are pulling on the chromosomes are shortening, the microtubules that aren't attached to the chromosomes are getting longer. They're spreading the cell apart further and further. Okay? So each chromatid is now an individual chromosome that will belong to a new daughter cell. Anaphase ends when the chromosomes stop moving. Okay? I only have one feature here that I'm concerned about. The duplicated chromosomes are separated, and the individual chromosomes are now moving into the daughter cells. The spindle fibers are shortening, and the cytoplasm is elongating. That's what's happening here. You can see that. See, as these cells are, or as these chromosomes are being pulled apart, the cell itself is beginning to elongate. And then finally, we get telophase. The word telo means the end. Okay? Literally in Greek, it means the end of times. Okay? So a discussion of the end times is telology. The study of the end times is telology. But the word telo means the end. Okay? So the last step in the nucleus splitting is telophase. The chromosomes begin to disperse. The nuclear envelope begins to reform. I reverse everything I did in prophase, right? That's all I'm doing. I don't really need to memorize a lot of stuff here. Everything that's occurring just makes sense in terms of what needs to be accomplished. And then usually at the same time, the nucleus has divided, mitosis is complete, but cell division, or the M phase, is not complete. The cytoplasm also has to divide. This is cytokinesis. This usually occurs at the same time, or at least begins at the same time as telophase. In animal cells, I start to get, and this is really more of a plant cell that it's kind of showing here, but in animal cells, what starts to happen is that I have some contractile proteins. What's the word contractile mean? It contracts. It closes in on itself, right? So I have some proteins in the cytoplasm here that are attached to the nuclear membrane, or the cell membrane, and they contract. They pull the cytoplasm inward. So I get a pinch of cytoplasm at both sides here as these contractile proteins pull the cell membrane towards one side or the other. They start to pull on the cell membrane here, and it starts to pinch off like this, and I get two cells eventually that are separated in that way. Okay? So in animal cells, this occurs by drawing into the cell membrane by contractile proteins until the cytoplasm is basically pinched in half. Okay? Okay. So those are the basics. And that is probably what you all learned in biology or AP biology or some other course, right? You've learned this maybe 2 or 3 times, and that's where it's probably ended. But this is a physiology class, and we want to know why all this stuff happens. We don't want to just know that it does happen. We want to know why this is happening as well. And there is an extraordinary amount of cancer biology wrapped up in all of this. So it's important for you to understand some of this because it starts to help you understand the very basic foundation of why people get cancer. And let me just pause for a second here to... And if you're interested in medicine at all, if you're interested in cancer, this stuff should really make you fairly excited to kind of learn more about cancer medicine. Okay? So we have to control this process. We can't let this process run amok. Okay? We know something about the cell cycle. We know what determines what it is that determines whether or not a cell divides. We know something about the cell cycle. What is it that determines? Now that we know, now I can't even read my own statements here. Now that we know something about the cell cycle, what is it that determines whether or not a cell divides? And I guess I should have put a question mark there in order to help me understand that. So we've said before, and we made a big deal out of this, and different cell types like skin cells have a tendency to constantly be dividing, right? I'm losing millions of skin cells every day. They slough off, they fall off. I scratch them off. I just scratched off 100,000 skin cells right there by doing that. Okay? I've got to replace those skin cells. You replace your entire skin surface every 4 or 5 days. Okay, so I have a lot of cells dividing. Okay, we also need to have cells that undergo cell division when the organism is growing or healing. But what did we say that made us at risk for? What did we say the fact that the cell is constantly dividing means as far as... Okay, but what? How did that happen? What did the? Yeah. Okay, so who's talking here? I can't see who's talking. Okay, so you guys are talking. Were you talking too? Okay, so It's really hard to tell who's talking when the masks are on. But we're going to get errors in our duplication of our DNA, right? And what we found out is that in most cell divisions, I said it's got to be perfect. In most cell divisions, there's anywhere from 1 to 3 or 4 mistakes. Okay, there's anywhere from 1 to 3 or 4 mistakes. But think about it. I'm duplicating 3 billion base pairs in 3 or 4 hours. If I only make 1 or 2 or 3 mistakes in that time, that's pretty good, isn't it? 1 out of 3 billion is a pretty low rate of mistakes. But if I don't fix that mistake, then that mistake gets passed on to the next cell, and passed on to the next cell, and passed on to the next cell. And then if I make another mistake that doesn't get fixed, now I have 2 mistakes that get passed on and get passed on, and then I have more mistakes that get passed on. And what I've told you is that we're really good at fixing our mistakes. In fact, when the DNA is duplicated, so if I have base pairs here, so if I have, let's say, adenine and thymine, and you don't need to write this down, and I have cytosine and another cytosine and guanine. Who remembers what pairs with adenine?
Thymine.
Thymine pairs with adenine, right? So I would put a thymine there, and that means also that adenine pairs with thymine and guanine pairs with cytosine. And so let's say that instead of the guanine there, that I got an adenine that accidentally paired there. The DNA, the protein that places these new base pairs in place here actually backs up and checks. It actually backs up and checks and says, hey, did I put the right thing there? And it can recognize that it didn't. And it can go back and actually put the right thing in. Okay? That's incredible, isn't it? It actually checks each one of these things as it does that, and it tries to put the right thing back in. But even if I still get one of these things that make it through, I have other repair mechanisms that go back and repair that. We're constantly checking our DNA to make sure that it's correct. And I have other repair mechanisms. And so very, very few of these things get passed on. But if I'm doing this millions of times a day, if I'm dividing millions of cells a day because I lose millions of cells a day, it's inevitable that I'm going to have some of these cells that get through with mistakes. And over the long course of time, over 30 or 40 years, I'm going to accumulate enough genetic damage maybe to develop cancer. But what's even worse is if I thwart my repair mechanism's abilities to do this. If I go out and I spend 5 hours a week in a tanning bed, or if I constantly am out in the sun without protection, or if I am constantly exposed to viruses that cause cancer because of the way I do things, or if I smoke, I am going to set myself up for these genetic mistakes because I thwart the repair mechanisms. All of these things, their main action is to break down my repair mechanisms. Okay? So, and it's not a judgment. I don't really care if you go out and smoke. I'd rather you don't. I don't care if you go out and tan. I would rather you don't. But if you do, that's your decision. I'm not going to pass judgment on you for it. It's just the way it is. It's biology, plain and simple. If you do these things, bad things will ultimately happen. Okay? So, no judgment. Just the way it is. So, because I've constantly got this, it should make sense that cell division has to be highly regulated. I have to control everything I do. I need for my cells to know when to stop dividing, because when they don't stop dividing when they're supposed to, that's cancer. or it's at least precancer. Okay? They have to stop dividing either because it's simply no longer necessary or because cell's genetic material has become damaged and needs to be repaired. The first thing I have to do in order to repair my DNA is stop the cell cycle. Right? I don't want that genetic damage to get passed on or passed on or passed on. I have to stop the cell cycle, so I have to be able to control this. It would be a good idea to allow genetic damage. It would not be a good idea to allow genetic damage to be passed on. When a cell slips the constraints of the controls that normally regulate cell division, groups of cells develop that can outgrow or have a selective advantage over neighboring cells, and cancer can develop. So, how does this work? In the 1970s, investigators discovered that if you fused cells in G1 phase with cells in either S or M phase, the cells in G1 phase immediately entered the S phase. So what do I kind of mean by this? If I have a cell that's in G1 phase here, and I make it so that the cell membranes of another cell fuse with the cell membrane of this cell, the cytoplasmic contents of this cell now have access to this cell, and this combined cell immediately enters the other phase. So this G1 cell will now enter the S phase. Okay? What do you think might be a conclusion that I would draw from this kind of an experiment? What might I say that this suggests? What might I say that that suggests? If I fuse this cell with this cell, and this cell immediately enters the S phase, what might it suggest happened? Think biologically here. Think outside your boxes. What might have happened here? No one? No one wants to hazard a guess?
The recovery nucleus was like shared with the S phase, and then you wanted to have to recover.
Okay, but what do you think? What do you think made that happen? What is there? Is there something about the fact that I've got this cell now in contact with this cell that could have made this cell to start going into the S phase? What's that? Well, it is going to speed up cell division, but wouldn't it suggest that maybe there's a chemical in here that got into here that made this one go into S phase? Okay, so if I do this kind of an experiment where I fuse this cell with this cell, and maybe that's what you're trying to say, maybe a chemical in this phase got into this phase and made this cell start to go into the synthesis phase. So the conclusion that we can draw from this is that a substance in the cytoplasm of the more advanced cell made the less advanced cell advance. Okay, something in here said, okay, you need to start doing what I'm doing. Okay? So we're going to find out that we have chemicals in the cells that are in the more advanced phases that can affect what's going on in other cell types. We have since learned that there are several checkpoints in the cell cycle that are controlled by cell signals, or signals both from within the cell and outside the cell. So I've got checkpoints. And if I get the correct signal, I'm going to advance. If I don't get the correct signal, I'm going to stop. Cell signals from within the cell are those that confirm that the cell to the cell, sorry about that, that certain processes for progression of the cell cycle to occur have in fact occurred. Okay? I am going to tell the cell, hey, I'm done with everything. I'm done recovering, right? In G1, I'm recovering, and I'm accomplishing the function that I'm supposed to accomplish. But now I've done that. I've been there. I've done that. It's okay for you to advance to the next stage. Okay, so cell signals from within the cell are those that confirm to the cell that certain processes necessary for progression have occurred. Signals from outside the cell confirm the fact that adding additional cells to the surrounding environment is necessary. Okay, if I've gouged out a big section of my skin because I ran my hand through a glass window or something like that, the outside environment of that wound is telling me that I need to add more cells, right? I've got to have more cells come into my skin surface. 3 major checkpoints occur, one each in G1, G2, and M phase. Okay? Depending on the needs of the cell and its functions, cell division is a highly regulated process controlled by a series of chemical checkpoints called a, what was the term I wanted? Called a, a cell cycle control system. Cell cycle control system. Okay, that's kind of an official term, the cell cycle control system. Alteration of chemical pathways through genetic mutation of involved proteins play key roles in the development of cancer. If I screw this up, there are hundreds and hundreds of proteins involved in this. And if one of those is not correct or it doesn't work correctly, I can develop cancer. Okay? So this is a very, very highly regulated kind of situation. Okay? I know there's still quite a little bit of time left in class. There's like 13 minutes left. I want to stop here because I don't really want to get any more involved in this right now because you've already had enough information for one day.
Thank you.
Okay, so find something to do for the last 13 minutes of class. Okay, I didn't think I was going to get quite this far that fast.
So here's the door.